Hello students, welcome back to our channel Diksha Karnataka. So your COMEDK exam is approaching, so we have started a series where we are discussing the top 30 questions from all the high weightage topics. So in today's video, we'll be discussing the top 30 questions from the chapter Biomolecules. Okay, so this chapter is very easy and scoring, and you just need to know some like facts about biomolecules, then it is a very high scoring topic. So if you are able to solve these questions, like you can easily expect 3 to 4 questions from this chapter. So if you are able to solve these 30 questions, you can like assure yourself that you are ready for this chapter for your COMEDK examination, right? So let us start with the first question here. So the first question is about study the structure of maltose and mark the incorrect statement. Okay, so this chapter from carbohydrates especially you have to focus on disaccharides and polysaccharides and the linkage between them, like which carbon is connected to which carbon. So many questions have been asked from that topic also. Now coming to this one, maltose is a composed of 2 alpha D glucose units. This statement is correct. Now C1 of one glucose is linked to C4 of the another unit. So you can see how we can say that it is both alpha group. You can see OH should be present here and OH should present here. This is alpha group, right? Now if you see next if you see C1 of one glucose is connected to C4. So this is carbon 4, this is carbon 1, this is also correct. It is a non-reducing sugar. So this statement is incorrect. So you have to remember, if there is any hemiacetal group, okay, if there is hemiacetal group, then it will be a reducing sugar. Hemiacetal group means it should have COH and OR group, right? Now if you see in this structure, this carbon, right, this carbon has one OH group and on other side it has OR group. So it is a hemiacetal group. So it will be a reducing sugar. So the incorrect statement for this one is option number C. Okay. And you can also see it is a disaccharide because the number of correct answer is option number C. It is a disaccharide because 2 monomer units are there. Okay. Now going to the next question. In DNA, the complementary bases are. So this is very easy question. AT and GC, that is adenine and thymine, guanine and adenine and thymine and guanine and cytosine. So correct answer for this question is option number B. Next, RNA is different from DNA because RNA contains. So we have 3 things in DNA and RNA. In DNA, so in the like nucleic acid, we have 3 things, that is sugar, nitrogenous base and phosphate group, right? Now if you see sugar in DNA is which one? That is your deoxy sugar, deoxyribose sugar. In RNA this is ribose sugar. Okay, now if you see the nitrogenous bases here are A, T, G and C. Here the nitrogenous bases are A, U, G and C. Then here it is phosphate group and here it is phosphate group. Okay, so now if you see the RNA is different, in the ribose sugar is different and the nitrogenous bases is different, right? So we have deoxyribose. So RNA is different from DNA because RNA contains ribose sugar and uracil. Okay, so difference you can see AGC is common, T and U are different. So the correct answer for this question is option number B. Now let us see the next question. In nucleic acids, the sequence is, so it presents like this. We have a sugar phosphate and we have nitrogenous base and we have a phosphate group here, right? So the order you can say we have base, sugar and then phosphate group. Okay, so the correct answer for this question is option number B. Now coming to next question. In cellulose, the D-glucose units are joined by. So you should know there are the 3 polysaccharides, starch, cellulose and glycogen. So you have in cellulose beta 1,4 glycosidic linkage. Correct answer is option number C. Now next, glucose reacts with acetyl chloride to form penta-acetyl glucose. It indicates the presence of. So this question is very important. There are 6 reactions you must remember that is used to determine the structure of glucose, right? So one of the reaction is which says that there are 5 OH groups in the glucose molecule. That is this reaction. Okay, so this 5 OH group is the correct answer for this question. That is option number D. Next one, in sucrose, the glycosidic linkage is between. So it is in sucrose, you know, there is the monomer units are glucose and fructose. Okay, so we have alpha and beta. So alpha 1 beta 2 linkage. So correct answer is option number D. Now coming to next question, what is the basic formula of starch? So we know starch is a polymer of glucose unit. So glucose is C6H12O6, right? Then here from what happens from the two glucose units, like you have one glucose units here and you have another glucose unit here. So from both of them, what happens? Water is reduced, right? So you will have, if water is reduced, we'll get C6H10O5, right? So the monomer of any starch is this unit, which is repeated many times. So the correct answer for this question will be option number B. Again, an easy one. Next, the protein responsible for blood clotting is. So this is little application based questions. So you must be knowing that fibrinogen is used for the blood clotting. Okay, correct answer is option number D. Next, which of the following is not a steroid hormone? So this question also, you might not be knowing the answer, but you can guess the answer, right? Oxytocin, you know, this one is not a steroid hormone. Steroid hormones are those hormones which have many heteronucleic cyclic big compounds are present. So correct answer for this question is option number C. So this one I can say it can be guessed, okay? Next, which of the following proteins is a transporting agent? Again, if you don't know, but we all know the function of hemoglobin. It transports oxygen from your lungs to the cells. So that is why transporting agent is option number C, that is hemoglobin. Very easy one. A non-reducing sugar found in honey is, okay, so this option was actually wrong. It is sucrose. So now if you see, fructose, maltose are all reducing sugar. You do not know anything about this. Maybe many of you may not be knowing about this sugar, but we all know that sucrose is a non-reducing sugar. So correct answer is option number A. Next, to which category of compounds do immunoglobulins belong? So again, if you see immunoglobulins, this is also a question that can be guessed easily. Immunoglobulins related to immunity, and we know antibodies are there for immunity. Correct answer will be option number C. Okay. Next one, ninhydrin test is used for the identification of. So this is again from the test of carbohydrates, proteins, and this one. So ninhydrin test is used for the prediction of amino acid. That is option number B. So all are very fact-based questions. So thymine, if you have seen the structure of nitrogenous bases, the methyl group of the fifth position of uracil is replaced with a methyl group. So that is why it is called as 5-methyl uracil. Okay, correct answer is option number A. Next, sulfur containing amino acid is again cysteine. So correct answer is option number D. Here I cannot, you have to just remember cysteine is the only sulfur containing amino acid. So such fact based questions are asked from biomolecules. So prepare the structures. You do not remember the structures, but some basic idea which are essential, non-essential amino acids, you can just keep in mind for them. Okay. So a little difficult for the students who haven't taken bio, but this chapter is highly scoring. At least try to complete the basic part, whatever is given in NCERT. That will help you to score most of the questions. Okay. Next, which of the following ions can cause the coagulation of protein? So again, coagulation of protein is caused by sodium ion. So correct answer is option number B. Next, burette test is answered by. Okay. So if you see here, burette test is a test again for proteins. So correct answer for this question is option number A. Okay. Next, you have indigestible carbohydrate, which is also a constituent of our diet is. So diet, cellulose is the polysaccharide, which is not digestible, but we eat it. So the correct answer for this question is option number B. Next, the sequence of bases on RNA molecules synthesized on the GCATS strand of DNA. So this we know complementary bases are there. So A with T and G with C. In place of T in case of RNA we will get U. So for G we will get C, then for C we will get G, for A you will get again U, then T will get A, and again for this one we will get U. So C, G, U, A, U, correct answer is option number A. Next, alpha helix and beta pleated sheets of proteins are classified as. So in the secondary structure it is there. So direct fact based questions. Correct answer is option number B. Next, which statement is not true for describing enzymes? So you must remember enzymes are proteins. Okay. So they are polypeptides. That is correct. They are glycosidic linkages between the molecules. So this one you can say if proteins are there, then what is the linkage between the proteins? Peptide bond is the linkage, right? So that is why this one is the incorrect statement. Correct answer is option number B. Okay, they are specific for a particular reaction, yes, and they catalyze the biological reactions. These enzymes are called as biocatalyst. Okay. Next, which of the following are monosaccharides? So very easy. Maltose is a disaccharide. This option cannot be there. Sucrose is a disaccharide. We cannot wear. Glucose and ribose are monosaccharides. That is option number C. Next, in nucleic acids, the nucleotides are linked by. So again, nucleic acids, the nucleotides are linked by your phosphodiester linkage, or we can say a ester linkage. Correct answer is option number A. So here I just want to revise. In carbohydrates, okay, the linkage is your glycosidic bond, and in case of amino acids, or we can say proteins, it is peptide bond, okay, and then in case of nucleic acids or nucleotides, it is your phosphodiester linkage, okay, phosphodiester linkage. Okay. Now let us see the next question. Amino acids exist as Zwitter ion. So what are Zwitter ions? Zwitter ions are those, like, what is the general structure of a protein? We have one NH2, we have COOH group, then we have R group, and then we have H group, right? In Zwitter ion, what happens in a particular pH, COH can give you an acidic like H plus, and NH2 can accept one H plus, and the molecule exists in this form: NH3 plus, COO minus, R and H. Okay, so this structure of the protein is called as your Zwitter ion, and this exists at a particular pH only. That pH value or that pH is called as your isoelectric pH, and in this position it will not move towards any of the cathode or anode. Okay, it will not migrate because it is a neutral molecule. It is a dipolar molecule, but it is a neutral molecule. So this pH value is called as your isoelectric point or isoelectric pH. Correct answer is option number D. Next, in DNA, the linkages between different nitrogenous bases are okay. So this question is little confusing. You may see phosphate linkage or something, but nitrogenous bases are linked by hydrogen bond, right? Between A and T, there is hydrogen bond. Between G and C also there is hydrogen bond. So correct answer of this question is option number B. Next, in Mollis test reagent, the substance used is, so Mollis test is used again for sugar, right? So that is used here is alpha naphthalon in alcohol. That is correct answer is option number B. Next, we have which of the following is not a protein? So all of you know cellulose is a polysaccharide. Correct answer is option number B. Then proteins can be hydrolyzed by using. So we can use acids, we can use alkalis, we can use enzymes. So anything can be used to hydrolyze the proteins or break the molecule. So correct answer is option number D. Now a nucleoside on hydrolysis. So this is very important. See, we have so first of all, if we talk about nucleic acids, okay, just understand the hierarchy here. Nucleic acids are made up of nucleotides, right? Now nucleotides are made up of sugar plus nitrogenous base plus phosphate group, right? Now this sugar plus nitrogenous base is called as your nucleoside. Okay. Now if you see here, what is the question is asking? If you hydrolyze a nucleoside, what are the products you will get? So that is your sugar and nitrogenous base. So if you see here, first of all, in the first option, heterocyclic base is given. That is correct. And orthophosphoric acid. So but there is no acid here, so that option is incorrect. Now if you see here, an aldopentose, that is the sugar. Heterocyclic nitrogenous base, that is correct, but orthophosphoric acid will not come in nitrogenous base. If the question would have been a nucleotide, then option B would have been the correct answer. Now if you see the C option, aldopentose, so there is a sugar here and a nitrogenous base also here, right? So that we'll be getting these two if we hydrolyze a nucleoside. So correct answer for this question is option number C. Okay. Okay. So students, these were the 30 questions from your biomolecules chapter. So some of these questions are from previous year questions. So some questions are very fact based that you may not guess in the answer. So that is why you have to practice more questions from this chapter. But the basic things like what you can say, the monosaccharides, disaccharides, their monomer units, their connecting units, okay, then reducing sugar, non-reducing sugar, essential amino acids, non-essential amino acids, such things which are already there in the NCERT, prepare them properly, and the vitamins and their deficiency. Okay, these are very important questions from this chapter. So I hope this chapter is, if you have any doubt in this chapter or any of the questions you are having doubt, please comment down. We'll discuss in my next video. And if you have found this video helpful, please like and share and subscribe our channel. We are bringing a lot of videos for your COMEDK preparation. So that's all. Thank you, and just best of luck for your exam.